Hello, welcome to the IBN laboratory tour. Inorganic biochemistry and NMR laboratory is devoted to the molecular understanding of the electron transfer processes performed by metalloproteins. Metalloproteins, in particular, multi-heme C-type cytochromes, are key players in the extracellular electron transfer performed by electroactive organisms that can be used in sustainable biotechnological processes such as bioelectrochemical systems for the production of energy added value compounds as well as bioremediation. To study these proteins, we use molecular biology tools to clone or modify them and microbiology and chromatography techniques to produce and purify the proteins. And to analyze their functional and structural properties, we use different spectroscopic techniques, in particular nuclear magnetic resonance and UV visible spectroscopy.
Hello, my name is Ana Silva and I am a PhD student at the IBN laboratory. In my case, I study how these metalloproteins that are so important for the extracellular electron transfer are made. What I study are maturation systems. Maturation systems are required for the formation of these proteins as the covalent bond that characterizes them between the heme and the apoprotein is not spontaneous in nature. Different organisms use different maturation systems to produce their cytochromes, and I'm interested in both the bacterial and the eukaryotic systems.
Hello, my name is Ricardo Soares and I'm a PhD student at IBN Laboratory. My project aims to understand how an anaerobic and marine bacterium called Sulforamonas acetooxidans is able to respire insoluble electron acceptors. One major limitation in microbial fuel cell technology is that the currently used bacteria are not allophilic. This means that they cannot sustain an environment with high salt concentrations. These type of environments are ideal for current generation because it offers high electrical conductivity. My work is devoted in identifying C-type cytochromes that are involved in extracellular electron transfer process and characterize them in terms of kinetics and thermodynamics of electron transfer and in terms of structure.
Did you know that electroactive organisms can also receive electrons from the cathode? I am Anaísa, and in my PhD project I want to study the processes by which sideroxidans litotróficos can receive electrons and use them for their metabolism. Sideroxidans litotróficos is an iron oxidizing bacterium that is able to couple the oxidation of iron by fixing CO2. And as observed for other electroactive organisms, during extracellular electron transfer, sideroxidase litotróficos uses several multi-heme C-type cytochromes to mediate electron transfer from the cell surface to the inner membrane. And with my PhD project, I pretend to purify and characterize all the proteins that are involved in this pathway.
Hi, I'm Bruno, and I'm a postdoc in the group. Although understanding the electron transfer process performed by the proteins is very important to unravel their mode of action, we also need to elucidate how they interact and transfer electrons with their physiological partners. This can be done using NMR, where you can see how the signals belonging to one protein shift when it interacts with a partner, indicating not only that they interact, but also revealing which protein area is involved in the establishment of this interaction.
The knowledge that we obtain for the electron transfer processes of these proteins can be used to modify and improve them. As an assistant researcher at IBN, my aim is to tailor electroactive organisms to make them better catalysts in bioelectrochemical systems. This can be achieved with rational manipulation of the multi-heme C-type cytochromes or by using directed evolution, where random mutations can be inserted in the proteins. With in vivo studies, the most promising proteins can be identified, and their study will allow to obtain information about the factors that limit the electron transfer in living cells, a knowledge that is still missing and is fundamental to optimize electroactive organisms to start using them in practical applications.
What if I told you that you can also shape microbial fuel cells for wastewater treatment and seawater desalination while producing energy? My name is Naswa, and here at IBN, I work on development of optimized microbial fuel cell for sustainable application. And how am I doing that? Well, on one hand, I modify the electrodes to make them more suitable for bacterial communities to attach on. And on the other hand, I tune molecular properties of the proteins exposed at the surface of electroactive bacteria so they can transfer electrons to the electrode more efficiently. Basically, I'm promoting sustainable match solutions so we can fight potable water shortage. So, are you up to the challenge?
Hello everyone. My name is Inês Trindade and I'm a PhD student at Inorganic Biochemistry and NMR Lab. My work is focused on study how bacteria acquire iron. I'll focus on Shevanella, the main organism of the lab, since for building so many multi-heme cytochromes, Shevanella requires higher amounts of iron than most bacteria. One of the ways that Shevanella does it is through the use of siderophores, small molecules that have a high affinity for iron. I study how iron is released from these compounds, and this occurs via the enzymatic action of proteins, which are called siderophore interacting proteins.
And did you know that Shevanella can also be pathogenic? I am Maria, and I am a master student in Master of Clinical Microbiology and Emergent Infection Diseases in the University of Lisbon, and I am focusing on Shevanella emergent infections. Some species of Shevanella genus, like Shevanella algae and Shevanella putrefaciens, are infectious, mostly causing soft tissues infection in human and fish infection in aquacultures. My aim is to identify genes that are considered virulence factors of these species. Virulence factors are the mechanism that bacteria have to fight against the host cells, and without them is expected the decrease of virulence. For that, I will use gene deletion techniques to construct the knockout strains and test them with pathogenicity studies in vivo, using embryos of zebrafish, and see if the gene influenced the bacterial virulence or not. I hope you are enjoying this lab visit.
In order to give you a brief flavor of the working conditions, here is a quick tour of the premises of the laboratory. I am also responsible for the NMR Center at ITQB, but this was not visited, but you are welcome to visit in person. Good morning. My name is Ricardo Louro. I'm leader of the Inorganic Biochemistry and NMR Laboratory, and I'm going to make a brief tour in the context of the work for biological and chemical remediation class of the Masters in Biotechnology for Sustainability. So our laboratory is a laboratory of two sets of benches. All benches are equipped with the necessary materials for biological and biochemical work. We have a glove box that we use either to grow bacteria that are not using oxygen or to do analysis of their bioelectrochemical capabilities in the context of understanding of how they produce electricity in the systems that we develop. We have obviously a fumes hood for dangerous work. We have growth cabinets for the cells. We have normal spectrophotometers for the analysis of biochemical samples. The labs are all empty today so that the tour can proceed normally. We have also a room for equipment where we have this cold chamber where we keep our samples and we keep the cells while we are analyzing it. The typical equipment for protein biochemistry, gels and such, benchtop centrifuges, pH electrodes to analyze the samples. We also have equipment for multiplate reading of growth conditions so that we can analyze multiple conditions in one go. Benchtop centrifuge, the molecular biology equipment that we also use to make mutants, and we use also as room with equipment for bioelectrochemistry so that we can actually analyze both the proteins and the cells, the organisms, when they are producing electricity. So here it is with potentiostats and multipotentiostats, again to analyze multiple growth conditions. We have also cabinets for growth and equipment for protein purification, so we are well equipped to do the kind of work that we propose in our lab. Thank you very much. So, you have seen a relaxed presentation of the fundamental research done in a laboratory related with health and sustainability, and it's work that we do with the ambition to contribute to the greatest challenge of our time, which is global climate change. If this line of research looks interesting for you, you are welcome to talk with the people in the lab or myself to discuss the possibility of joining the team.